Hello everyone. Now, as we remember in our last lecture, we prepared our first RNA-seq pipeline. So here I'm on the TBioInfo platform, and I can see the first pipeline right here. So let's take a look at what is inside. If I click on the link, I can see here download pipeline output files. Let's just quickly review what exactly we did. So as you remember, we found this data set from a publication that has cell lines, cancer cell lines, and each cell line belongs to a certain class, a certain group of breast cancers. Now we took all of the data, raw FASTQ files, and we started processing those. So we uploaded the files, the raw FASTQ files, and then we removed some of the adapters and the PCR amplification. And then we mapped on known transcripts using bowtie2t and produced an RSEM expression table. So what we should expect here is the RSEM expression table that represents gene expression and isoform expression from the cell lines that we are studying. Let's take a look under Download Output Files. And now you can see here several files. So you have expression genes, FPKM not filtered, just FPKM, and then not filtered, this is going to be in counts, and expression genes. So all of this belongs to gene expression, and then all of these belong to your isoform expression. There's also going to be a stats table that shows you how many of the reads were mapped, how many of the reads were not mapped to anything. So this is an interesting file to take a look at, but today we're going to look at the gene expression table and try to understand what insight can we gain from looking at this file. So let's download expression genes FPKM, which is going to be a TXT file, but of course you can take this file once it's fully downloaded and you can open it, for example, in Excel. Okay, and now let's take a look at what's inside. So we have gene names. ENSG stands for ensemble, G stands for gene, and so this is an ensemble ID. I can, for example, take this gene and I can go to Google and search for it in Google. Let's see what I will find. Okay, so this gene, you can see right here is T-span6. Okay, so some gene, and I can read about what it does. Now let's go back and just see what are the column names. Now the column names are actually the samples. If you remember when we downloaded our FASTQ files, each one had an SRA read. So this is sequence read, SRR. So now we're going to, and now we just added no adapters, no PCR parent. So these are some additional metadata that we know what happened to the files. But it's going to interfere with what we're trying to do, so let's quickly clean this table up by removing these additional things for all of the samples. And what we can see here is now clearly that for every cell line, every sample, we will have a gene expression value. How many genes are expressed? If we go all the way to the bottom, we can see that there's about 20,500 genes. What can we know about 20,500 genes across these samples? Let's take a look at one of the genes. So here's one gene. Let's just insert here quickly a bar chart, and let's take a look at what this could mean to us. Okay, so first of all, we can see that a number of samples have higher expression. Some of them have distinctly lower expression. And then we have all kinds of in-between values. So it's not very informative, but also we don't know what these SRRs mean. So let's go ahead and find out what are these SRRs. So the table that I shared with you actually separates these SRRs into two subtypes that we have, the normal-like and the cloud and low. Now let's take a look at how they separate. So again, if we take the first gene and we take a look, we see that, interestingly, there's a normal-like that is low and a Claudin-low that is low. And then we have these different groups. It looks like normal-like maybe is a little bit less expressed than the Claudin-low. So this, however, is not very informative, right? Because I would have to go through all of these different genes and look at, okay, well, what does this gene do? What can I learn from this gene? How does it maybe associated with Claudin-low or with normal-like? It's very hard to understand something from so many. There are several things that I can do here. The first thing, as we saw in the previous lectures, we have only two groups, so we can use student's test or t-test to identify those genes that definitely have a difference, a significant enough difference. But there are other techniques as well. So today we're going to look at PCA, which is principal component analysis. It's a dimensionality reduction technique. And then we will take a look at clustering techniques, several clustering techniques that we have on the platform, just to see what can we infer from the data that we have in these tables. Now with two groups, it should be fairly straightforward. Let's take a look. So I have under areas of analysis, supervised and unsupervised data mining sections. So let's take a look at the table that we have. We have this gene expression table that we filtered already. Let's choose it. It already has annotation what group each sample belongs to. And let's just quickly prepare a PCA of the table that we have now. What does PCA do? Principal component analysis. It will use an orthogonal transformation to convert a set of observations, in our case, these are different samples, into a set of values of linearly uncorrelated variables called principal components. So what it will do for us, it will plot the gene profiles and generate a visual graph where it's easy to see how these groups are associated, closer together, or unassociated, so separated. Okay, so let's take a look, and this is two groups, right? So we have normal-like and Claudin-low. And our pipeline finished. Let's take a look what's inside. The first one that we get is this PCA plot PDF. We see here a good separation between Claudin-low and normal-like. Now another thing that we should pay attention to are these percentages. These are the weights of the principal components. The higher the percentage, the more data variance it explains. So which principal component here is more valuable to us, shows more separation? This PC1, because it shows us a clear separation and it's got more percentages here. Now this separation is not as informative, and you can see that the separation between on the y-axis is not as clear. So this was pretty easy, right? We have two types of cancer, and we can see that dimensionality reduction works well for these kind of simple data sets. Now there's a lot that we can improve, but let's take a look at a more realistic picture. What happens if we have many subtypes? What can we do then?